Okay, I'm going to invite all the speakers back up. Thank you. Okay, so for this portion, since we're tight on time, I'm just going to have the speakers briefly answer one question that I posed for them, and then we have like 4 questions that the audience posed that we can talk to together. I encourage even people in the audience, there's a microphone here that you can even answer some of the questions too, because we want to hear from all of you. So the question I have for you guys is, what is the best case scenario for integration in your particular perspective? As succinctly as possible, like 1 or 2 sentences.
We develop cure for all diseases.
Cool.
Obviously, data is always a problem, right? The best case scenario is we have all the data that we need, and we can compare all these things all at once, everything everywhere all at once.
Yeah, I'm with that answer as well. I think we'll have a very detailed understanding of how to bridge across scales and across, different biological phenomena.
Ready for a lot of insight into this answer. Just include a lot more rural people in our studies. Very straightforward. But I think ideally it would be based off of, if rural people can't participate, that it's not based off trust issues in science, and that it's more based off of, if we truly don't have the finances or means of including people.
Okay, cool. Thank you. So we have a lot of questions. Okay, a lot of these are directed for specific speakers, but I'll try to do a mix. But one of them that is interesting, I guess this is for Katie, but any of you guys can chime in the audience too. What do you think the future of multimodal studies should focus on?
Actually, that's quite an interesting question, because I think there are many directions one could go with that. I think, interpretability of the models that we use to combine modalities, I would say, is one thing that we could prioritize. I think we can gain a lot of predictive power by sometimes merging different modalities and information together. But at the same time, I think we want to keep a focus on how we can gain new knowledge from doing that and from seeing the connections across different modalities as well.
Does anyone in the audience, you don't have to answer, have any input? Okay, yeah.
Is it work? Yeah. Hi, I have a question for Brendan. So in your view, what are the main constraints or factors that would block data collection in rural areas in the US and in the world in general?
Yeah, I've thought about this probably more so towards the US. I would say the US is maybe a little more privileged in terms of more resource-limited settings. You might have an MRI within 10 hours. Most rural places in the United States, that distance is maybe 4 hours max, 3 hours max. But really, the majority of the rural population in the US lives closer to a metropolitan area. So in some ways, it may only be a few hours. And in those situations, I think that, sorry, can you repeat the question? I got trailed off there, sorry.
Just what are the main factors that would block data collection in the US or in the world?
Yes, so I think what I was basically saying, yeah. So there's scanners basically everywhere for the most part, at least within local hospitals. So very few situations in the United States do not have an MRI scanner within a hospital. It might necessarily be a research site. So I think it's kind of building the partnerships with those areas. And I was at the Rural Health Conference recently, and so if we have MRI scanners, they're fairly ubiquitous in the United States compared to elsewhere. How do we get people to do this? So what are the incentives that we might provide to the hospitals or to the patients? And a lot of people that I talk to, it's kind of more so, that we're not just coming in to observe, or we're from Yale and we're here to help and to learn about you. I think that we can kind of have some kind of value proposition that we're offering to the rural populations or rural hospitals in terms of what are they getting out of it. And I think that might come in kind of at the research question stage. what are the research questions that are important maybe to the rural communities or towards those hospitals that we can provide value instead of just kind of taking and learning or observing in a weird experimental way.
Thank you.
Can I just, because you've raised a really good point that it's important to have the connection to the rural populations, because I think we've been focusing a lot on the technological disadvantage that we have. I think at this stage, there's now low field MRI, it's very mobile, AI is being used to, revolutionize that field. So I think we can solve the technological part, but I think the connecting to the population is really important too.
Cool, so I'll ask 2 questions at once. They're for 2 different people. One of them is for Sadan. How can multi-scale integration truly work? Using different scales is nice, but how do you understand their interactions? And the other question is, I guess, to Brendan and James. So your approach to consider more differences kind of opposes James's approach to discover universal laws. Can you comment on that?
Sure. No, I agree that it can be hard to understand these things. Biophysical models help, right? They simulate interactions between these scales, and as we have better data across these scales, we can actually verify how valid these models are. Also, harmonization efforts, things like the NeuroMaps toolbox, where data from different scales are put into a space that can be kind of tested with general linear models and things that we're very familiar and comfortable with, I think can help you identify these things, at least in a linear way. I think what really helps in doing these sorts of analyses is to focus in on a clinical question. James might disagree, but like big brain principles are really important, but they're very untractable with the kinds of data I think that we currently have. So narrowing the state space and focusing on a disorder, you can understand it. We saw this recently with the development of muscarinic receptor drugs for schizophrenia. That came from multi-scale research, and it's led to drugs being approved that reduce suffering. So I think the proof is already here that we can do these sorts of analyses, and they do change lives.
I could say a little bit. So thinking of rural maybe as a covariate or something we want to remove from our analyses versus including it, I think that kind of as it relates to this, if we have social isolation or something like that, the relationship of that with the brain may actually be similar whether you're in an urban area or a rural area, but we might not be capturing the same amount of social isolation if we're only capturing urban participants. That's kind of one idea towards that. So those relationships might not be different, but we might be actually just not capturing the data. But I think another interesting part, particularly as it relates to brain behavior relationships and predictive modeling, there's a lot of emerging evidence that there might be subpopulations within predictive models or within brain behavior relationships where if we're predicting or just relating the brain to something like cognition, that the majority of people may have one way of getting there in their brain and kind of what underlies that inter-individual variation for cognition, but then maybe 20% or 10% or 5% of individuals have a different brain behavior association. So it's kind of in tension a little bit.
Cool. So this, does someone speak up?
I just want to say that finding using a multi-species approach to find generalized principle, it's not mutually exclusive to finding individual variability. I can see that there are environmental genetic factors that support individual variability, but I also want to highlight that there are certain things in the brain that are evolutionarily conserved, and there's a reason for that, right? Otherwise, the evolution would just remove those things. I can see Ian hiding in the audience. The cerebellum, for example, is one of the oldest structures in the brain. Why? We have very limited understanding of why. The cerebellum is very, very important in the brain, and we know that it has been there for a long, long time. So there must be a reason. And I think that's what I'm trying to find in terms of multi-species approach.
Yeah, sorry, these are just the questions I forgot to advance. You can vote on these also, the audience can. Oh, you have a question?
I was hoping, I thought those were wonderful answers, and I just wanted to follow up a little bit. I do think some of this is about, does anybody remember studying Tinbergen's scales, right? The 4 different levels of explanation. Part of this is that one of so these can both be true, right? That evolutionary answers are different than mechanistic answers. But do be careful, because it's not the case, right? That just because something is disadvantageous means that it will be selected against. That's just sort of not not true in evolution. But I think that there's another reason to study cross-species similarities and dissimilarities, right? One is to understand the evolution of the brain, as you're saying, to get at that. But the other is to effectively use animal models, right? So there's a whole bunch of things that we can do in animals that we can't do in people. And if we're going to actually advance, brain health, we're going to have accurate models in animals of what is and isn't the same across species. So it doesn't have to be universal. It could be just present in one animal model, right? It could just be, present in owls or something. And if it's present, if it operates that way, if the information is transmitted in a similar way, and we can target it with whatever device or drug, then it's useful, right? That's a comment, not a question in classic academic style.
Okay, so I'm gonna pose there's 3 questions at once, but 2 are for Katie. To Dr. Chang, among all physiological measures, to choose one to cover most, what would that be? And also, could you describe what kinds of information are missed when transforming data from EEG to fMRI and vice versa? And then for the whole panel, someone asked an interesting question about, can you speak on your own experiences with fostering collaborations?
So in terms of physiology, I don't know if I could pick, say that there is one that is more important, because I think each one gives us an important lens into the body and in brain-body interactions. I think most typically in fMRI studies, we tend to collect respiratory and kind of like a pulse ox signal, just because that's often most readily available with our scanners. But I think we also miss a lot by not kind of going out to see what we may be missing in measures such as I don't know, blood pressure, carbon dioxide. Now, these are much harder to collect, but in a way they can give us a lot more, additional quantitative information that can help us interpret the data and learn new things about brain and body. And so in terms of what information were missed when transforming EEG to fMRI, I think we are kind of still trying to figure this out. I think that the studies that have, emerging studies these days are showing that there's maybe a lot more shared than we may have recognized in the past, just by virtue of being able to, say, take a machine learning model and through some maybe complicated nonlinear transformation be able to reconstruct like a deep brain fMRI signal from scalp EEG, which I think also presents a lot of opportunities in terms of more accessible, low-cost imaging. But also, I think where people are still trying to probe, what are the limits of that? I think we can't reconstruct perfect spatial, high-resolution spatial information with EEG that we can get from fMRI, and vice versa. We are going to miss a lot in terms of certain EEG phenomena. But yeah, it's hard to give a quick answer on where we might be missing things.
But Katie, what's the minimum physiological signal that we should definitely measure all the time?
So, well, this is more from a data processing perspective, but I think that if you have a PPG signal, you can also extract respiration through some fancy processing. So I don't know if that's a cheating sort of answer, but yeah, then you can maybe derive more information. But we're, now this is a little more exploratory, but we find that we can extract more physiological information from bold fMRI than at least I thought was possible before. And I think it is because there is so much, shared fluctuations that if we kind of learn the transformation between these, we might be able to approximate really well, a respiratory signal from fMRI. Not perfectly, and I think a challenge of that is we're probably not using enough diverse data to be able to see all of the, cases to really be able to study individual variability. We have done a study on this in healthy controls, but, then the question is, well, can we actually can we apply it to diseases or to populations we have not, trained our models on? So I think it's more work to be done.
And then there was a question about collaborations, if any of you guys had experience with that. Can you speak on how to build them? And anyone in the audience too, if they've had experience also.
Nobody's ever collaborated up here, it seems.
I will talk about experience. With collaboration, you know, it can be hard to reach out to people who are in completely different fields, and often you won't get a response. And that's what I initially had when trying to delve into common molecular imaging. And it's kind of going opposite to what I said in my talk in terms of collaborate in large teams. So what I did first was to use tools already available like neuromaps and things like that that are based on normative populations to start incorporating that into my analysis to kind of show people that I had some understanding and interest across scales. So maybe starting in smaller teams and alone is okay to demonstrate some sort of efficacy and leaving your silo, and that actually opened the gates to a lot of collaborations with people who are experts in these scales, in other scales of imaging.
I think I'll share my personal experience. I'm a physicist, so how the heck am I doing species stuff? I think it started at OHBM, I'm pretty sure, because I was just walking around a poster, and I went to a poster that's completely irrelevant to what I'm doing, and it was an animal work, and that's kind of where my passion started, I think. So go to the poster hole and make sure you go to someone's posters that's not within your group or within your field, because you can learn so much more.
Okay, I'll have us end with this last question of how do we tackle differences in the quality of data collection when robust replication is challenging with large data sets?
I will say that you do not need large data sets for multi-scale research. You can have single subject data sets that have like very successfully shown integration. You know, the data set I showed with 90 people with mortem imaging and postmortem, you know, the effects they were finding were so large because they're tapping into real biological variability that you don't necessarily need larger scales. In terms of determining quality, once again, you have to trust collaborators and learn about the other scales that you're working with, I guess.
Cool. Are there any audience questions that anybody wants to answer? Or ask, sorry. No.
Do we have the question where we can see it here, Maya? Can you pull the question back up, or which number is it up here?
Which question?
Oh, the last one? Okay, yeah, differences in kind of data collection when robust replication is challenging. I think this makes me think towards the idea of, do we need a one-size-fits-all approach? So say with rural populations, do we need to include them in thinking towards maybe predictive modeling specifically with brain imaging? Do we want a predictive model that works well for everybody, but then you might suffer in terms of accuracy overall for everybody? And there's some trends towards creating population-specific models, and this doesn't have to be specific to necessarily predictive modeling, but to your point of kind of small data sets, if it works where you're using it, it might be beneficial. We don't always need things that generalize. If you're building a sex-specific model or an age-specific model, or looking at associations, maybe we don't need to worry about kind of the differences in data collection. And just for example, in rural areas, if you have older MRI scanners, they're low resolution, if we build a model at again, Yale or something, and it doesn't necessarily generalize to rural populations, that might be okay if it works well at Yale, but maybe we want a rural-specific model that's trained on maybe the lower kind of resolution MRI scanners.
Cool, thanks, Brendan. Before we end, sorry, how does this, I can't see the advancing. I can't see. Is it possible to advance on your end? Yeah, okay, cool. I want to give special thanks to our speakers and like Ashley Siegel for her support, and my advisor, Dustin Shinos in the front. It's also his birthday, so you can wish him a happy birthday. But yeah, for these people. We're also working on a, sorry, can you advance it again? Sorry. Okay, we're working on a kind of like a review / commentary on like multitudinal brain imaging, like integrating across domains, and we go a little deeper in the perspectives that each of them are representing. And we're interested in feedback since we all have our own like specialties and we don't know everything. And so we have like a preprint that you can read, and please reach out. My email is on there, but you can reach out to any of the speakers too if you have any other, further questions. But yeah, can you advance it one more time? Sorry. There's a survey for the that you guys can fill out. But yeah, thank you for coming and participating. Yeah. Thank you.